And right, here we go. I'll start the slides up. And let's talk about echinoderms, the spiny-skinned critters. Echinos, Greek for spine or thorn, and derm, Greek for skin. We've been talking about Bilateria, which is basically all of the animal kingdom that isn't a cnidarian, ctenophore, sponge, or placozoan. And I talked about how you can divide those into protostomes and deuterostomes. At least ancestrally, that depends on whether the little dent in the early embryo called the blastopore becomes the mouth, in which case it's a protostome, or the anus, in which case it becomes a deuterostome. Now, developmental patterns can vary so much, even within a phylum, that you don't always see this when you look at actual embryos. But the ancestral pattern seems to hold up pretty well, that all the critters we classify in the protostomia, many of them do develop this way, and they do seem to come from a single common ancestor. This seems to be a pretty good clade. Most of the class is going to be protostomia. Lophophorates feed with a ring of ciliated tentacles called a lophophore. There's 3 phyla in the Lophophorata, only one of which you could actually see in Arkansas, but they're still around. Platyzoa includes flatworms and some smaller critters that are pretty common around here, like rotifers and gastrotrix. Trochozoa includes the segmented worms and the mollusks, and mollusk is snails, clams, squid, slugs, and actually lots of others. And then finally, the ecdysozoa. Ecdysis is Greek for molting, and these are critters that molt their cuticle. The big ones there are nematodes, which is the roundworms, arthropods, which is crustaceans like squid and, not squid, come on, Wagner, shrimp and crabs and lobsters and crawfish and insects and spiders and scorpions, all of those things with jointed legs, which is what arthropoda means, and some others that we'll get to. Deuterostomia, mouth develops second, and the 3 phyla here are echinoderms, which we'll talk about today, hemichordates, not a very large phylum and not one that you could see in Arkansas, nonetheless an evolutionarily important one. And then chordates, which includes the urochordates, aka the tunicates and the salps, exclusively marine. Cephalochordates, which includes the amphioxus, which you're all going to be singing about. And then craniates includes hagfish, which are still technically not vertebrates because they don't have vertebrae as such. They've got a skull but no backbone. And then the true vertebrata includes everybody else, and we're not going to talk about them. You can take VertZo with Dr. McDonald if you're just dying to find out more about those guys. Right, so let's look at the echinoderms. Development is deuterostome. The blastopore, that dent, becomes the anus, not the mouth. The symmetry of adults is mostly radial, but we've got fossil evidence and developmental evidence that echinoderms did evolve from bilateral ancestors, and that radial symmetry probably reflects an evolutionary switch from, moving around actively like typical bilaterians do, to a time where the ancestors spent sitting on the sea floor, waving their arms in the air in that situation where they could receive stimuli from any direction, not just one. Remember, we talked about this when talking about the evolution of bilateral symmetry. All echinoderms, there might be a few that have lost this, and there's plenty that have reduced it, but bloody near all echinoderms have a skeleton inside the body covered with skin, so it's an endoskeleton, not an exoskeleton. The skeleton's made of calcium carbonate plates. The plates may be flat, they may be spines, they may take on other shapes. They can be adapted for various things. The fancy name for one of these plates is an ossicle. Os is Latin for bone. Ossicle means little bone, though they're not technically bones. They've got a type of microstructure that I'll show you in the next slide that's called stereom. And they are coelomate. They've got a large fluid-filled body cavity filling up a good chunk of the volume of the body. The coelom's divided into three. In embryo, they are basically front, middle, and rear divisions of the coelom, or protocoel, mesocoel, and metacoel, if you must. The actual body cavity comes from that posterior part of the coelom, the metacoel. The anterior division becomes a very small circulatory system. It's a few blood vessels, but it doesn't do very much. The middle division branches and wraps around itself and becomes a defining feature of the echinoderms, the water vascular system. In addition to the circulatory system, this is a second set of branching canals that conducts water through the body. And it's what enables echinoderms to move, usually, to feed, just generally get around. Very important for echinoderm life, this water vascular system. So let's look at the main features. That's a broken sea urchin plate, and you can see it almost looks foamy. Think of it like this. If you have a solid object and you start stressing it, you're going to put a crack in it. And if you keep stressing it, all of the force of that stress is concentrated right at the tip of the crack. Or it works the same way if you're tearing paper. Imagine you've got a small rip in a piece of paper and you keep pulling at it. All of that force is concentrated right at the tip of the tear, right? At the tip of the rip, as it were. And all of that force can act to tear the paper further, or break the plate or whatever it is that you're breaking further. You can stop a crack by drilling a hole at the very end of the crack. You're basically taking away that point where all the force is going to be concentrated. If any of you ever end up being, if any of you ever end up as a rock band drummer, one of the tips that drummers know, especially if you're poor and you can't afford to buy new gear, is that every so often one of the cymbals will show a crack. And if you can't afford new cymbals, but you can't, you don't want the cymbal to crack all the way through when you hit it, which it will eventually, you can just drill a hole right at the tip of the crack. And once you've taken away that little point where the crack was propagating, the crack can't get any longer. You basically remove that point where all of the force was concentrated. Your cymbal is still going to sound funny, but at least it's not going to get any worse. Now apply that to what you're seeing there, that microstructure of a sea urchin plate. It's got an awful lot of round holes in it. Frankly, it almost looks, like foam. In life, the holes would be filled with living tissue, but this works to stop crack propagation. If this thing starts cracking, the crack will hit one of these holes, and then there won't be a focal point at the very tip of the crack where all of the force can be concentrated. So the upshot is you can make a very strong structure this way, without having to spend metabolic energy to make it solid. You can make it light. You can make it efficient to grow without having to sacrifice the strength. So it's the same idea as, styrofoam, for example, which is fairly tough, despite being very light and being mostly filled with air. This is how you can make materials that are light but still strong. In some echinoderms, the ossicles butt together very tightly, almost like bricks. In others, like starfish, they may be a lot more flexible and the ossicles don't fit together nearly as tightly. Ossicles can be specialized in various ways, and in many echinoderm species, you have ossicles that form these pincher-like structures. So on the left, that's a pair of ossicles operated by some very small muscles. And those are often used for defense. If you're very slow, sometimes you might use these to defend against predators, but also, a lot of critters are going to try to grow on you if they can. The larvae of corals and other critters might try to settle down on your spines and grow there, and you can clean them off with these pedicellariae. There is at least one sea urchin that has been known to kill people with its pedicellaria. I think we've got a picture of it coming up, so I'll talk about that one in a bit. So the ossicles are one of the basic things about echinoderms. The other is the water vascular system, which comes from the left middle division of the coelom, the left mesocele, when the echinoderm is still an embryo. What it develops into is a set of branching canals that extend through the body. So in your typical starfish, you have a canal running around the gut in the central part of the body. It's called the ring canal. And then you have typically 5 branches running down each of the starfish's arms, called radial canals. I'll show you a diagram of that in a bit. And usually those radial canals extend into extensions which poke out of the body, called the podia, commonly known as tube feet. Depending on what echinoderm you're talking about, there may or may not be suckers at the tip of the tube feet. In many starfish, there are. In the sea urchins that hopefully I can show you tomorrow, there are suckers on the tube feet, but in some starfish and brittle stars, there aren't. So it depends on the taxon. These podia are frequently used in feeding. They're frequently used in locomotion. Starfish move on their tube feet, for example. They also play a role in gas exchange. Some carbon dioxide is given off and oxygen taken in through the tube feet. And the podia are arranged in long rows. And the word for a row is an ambulacrum. And why don't I just show you? There's the underside of a starfish arm. And running down the underside, you have a whole row of tube feet. It's actually 2 rows of tube feet, but they're all connected to that same internal radial canal. So that row of tube feet is called an ambulacrum. The podia, are the things sticking out of the underside that end in suckers. Not all echinoderms and not all starfish have podia that end in suckers, but many of them do. And the starfish can not only move, it can pick up prey items and pull them open using those podia. Starfish are very slow but very determined predators. Incidentally, I'll talk about these in a bit, but if you look at the upper surface of that starfish arm, you can see what looks like kind of grayish fuzz. Those are called dermal gills. They, in fact, connect to the coelom, and they're another means by which the starfish can do gas exchange. I'll show you those in a diagram that's coming up. And then the knobs and things are reflect the ossicles that are just under the skin. Right. That's what you'd see if you dissected a starfish. Water's taken in on the side away from the mouth, the side that's normally facing up, through a sieve-like structure called a madreporite. It's a specialized ossicle with a bunch of holes in it. Water passes through the so-called stone canal into a ring-shaped canal called, sensibly enough, the ring canal, kind of like a traffic roundabout, which, since you live in Conway, you know a lot about those now, those of you that are in Conway. And the ring canal branches into 5 radial canals, and each radial canal branches off and gives rise to a podium or tube foot. You can see that from each radial canal, you have a series of lateral canals. Each one leads to a tube foot, and the tube foot has an ampulla up at the top, kind of like the bulb of a medicine dropper, or the bulb of one of those plastic pipettes that you've got. Muscles in the ampulla can contract to force the tube foot to extend, or they can relax to allow the tube foot to contract. So these tube feet, again, act like hydrostatic skeletons, taking advantage of the fact that you can't compress water down to smaller volume. And if you compress one part of a fluid-filled structure, the other part has to expand. All right, connected to the ring canal is a so-called Polian vesicle, which I think is has to do with regulating the pressure of the entire system, if memory serves. Right, there's a slice through a starfish arm. The open space in the middle of the arm is called the paravisceral coelom. That's the main, coelom proper. And inside it, up at the top, you've got that pair of kind of crescent-y shaped, stripy-looking things. That sections through extensions of the stomach. That sections through the digestive cecae. The stomach is in the center of a starfish, but it's got these branching extensions that extend into the arms. And then below those, on the left and right, those 2 lumpy things are gonads. And the space that they're in is called the paravisceral coelom. And if you look at the dorsal surface, the top of the picture, those things that look like tweezers are pedicelaria. And you can see some extensions of the coelom that push out between the ossicles and make these little, lumpy extensions at about your 11 o'clock and your 1 o'clock. Those are papillae or dermal gills. Those are these extensions of the coelom that stick up. You can see they've got very thin tissue covering them, and these function in gas exchange. Sorry, something my wife was showing me. Right. If you look at the lower part, you can see a cross-section through a pair of podia, and each one's got the ampulla and then the tube foot sticking out of it. So that's your water vascular system. You can see they're both connected to a ring canal by lateral canals. And then below that, there's this dinky little heart-shaped thing. In the center of that is the nerve cord. There's no real brain, but there's a ring-shaped nerve cord and then radial nerves. And on either side of that, you've got the blood vessels. You've got the hemal system. And it's not very big, at least as far as gas exchange. A lot of echinoderms seem to get more of their oxygen through those papules and or through the water vascular system than they do from the hemal system. I think the vessels do pulse a little bit, but there isn't much of a heart. So anyway, there's all your three divisions of the coelom. And if I had the brains the gods gave cheese, I would have color-coded them before showing you this. C'est la vie. La vie. Right. More features of echinoderms. This is really cool. Actually, I had a good friend in grad school who had done some research on this. Starfish have connective tissue, like you do. And it, like all animals, it's got the protein collagen, or varieties of collagen. There's something like 30 different types. Just like your own tendons, for example, are mostly made of collagen, this tough, fibrous protein. Echinoderms make a type of collagen that is under neuronal control. The nervous system can actually make that collagen rigid or flexible. So the echinoderm's nervous system can control the rigidity of its own body wall. This is very useful for echinoderms that filter feed because they can extend their arms to catch food prey and then simply lock them in place. Just make their connective tissue turn rigid, and then they're not expending energy in holding those arms up, right? If you have to hold up your arms, your arms will get tired. But imagine if you could hold up your arms and then just simply freeze all of your joints into one solid bone. Then you wouldn't get tired any more than a tree gets tired holding up its branches, right? A tree's not exerting energy to hold its branches up, right? A tree is just that shape, and it'll hold its branches up unless they break. Echinoderms do something similar. They can simply make their connective tissue rigid and hold their arms up without getting tired. Starfish can pull prey apart, the clams that they feed on, in part because they can use that catch collagen tissue to lock their arms in position and not get tired holding them that way. So a very neat trick there. Many years ago, I came across a paper from a Japanese scientist who had actually written a song and published it in this scientific paper about the wonders of catch collagen. And I can't remember all of it, but it ended with, in autotomy, locomotion, posture, control, lovely catch collagen plays a key role. I need to find that. There was a very sweet, eccentric Japanese invertebrate biologist who used to write songs about echinoderms, and that was one of them. Right. The nervous system is basically a nerve ring with radial nerves, but there's no real brain. There's no excretory system that's specialized. I think waste is able to just diffuse out. Respiration goes through the podia and through those papillae that I showed you, those dermal gills. A few echinoderms can tolerate brackish water, but other than that, all species are marine. There are no freshwater echinoderms. You're never going to catch these in Arkansas And if you do, it's probably somebody's old wind chimes. There's a species of sea urchin that has very big spines that used to be collected and made into wind chimes and sold in souvenir shops in Hawaii. So if you find sea urchin spines in Arkansas, it's probably that. There aren't any living ones in this state. This is the largest phylum that is marine only. Most living echinoderms show almost perfect pentaradial symmetry. There may be a few things that are a bit off-center. The madreporite is usually a little bit off-center, for example, but it's pretty close. There's a couple of starfish that show it very nicely. Pentaradial, of course, means there are 5 angles that you could rotate this starfish through and get the same figure. And they're, 72 degrees, 144 degrees, and the others that I can't be bothered to calculate in my head right now. So there's 5 angles through which you could rotate that picture to get the same picture. There are some modern species that have modified their degree of radial symmetry just a bit. That's a species from the North Pacific, Pycnopodia helianthoides. All right, my wife is stepping out. I love you, sweetie. Have as good a time as possible. All right.
Your kids have said hi.
I shall. Yes, my wife sends her greetings. Right. This one's actually found on rocky coasts of Washington State, Oregon, British Columbia. It's the kind of thing you might see if you ever get to explore those states, if we ever get to travel again once the pandemic is over. Anyway, you can see that that one's modified their degree of radial symmetry just a bit, with probably about 16 arms. There's one that I have seen in Baja California, Mexico, that gets up to, I think, about 36 arms tops. So some of these are not pentaradial anymore. They've, added quite a bit of arms. I guess because there's an arms race on. And there's some species that have modified that pentaradial symmetry to bilateral. So on the left, there's an African species of sand dollar. You can see the 5 ambulacra up at the top, those 5 things that look like almost like flower petals. They're actually made up of a series of very fine holes in the skeleton. And those are almost perfectly pentaradial. They still look very much like a 5-pointed star. But you can see the overall shape of the skeleton is no longer pentaradial because, half of the sand dollar is divided up into those finger-like extensions. And then another species I used to see in Baja California, Encapi, the so-called arrowhead sand dollar. You can see that that one is bilateral now. You can barely see the ambulacra, but the 2 petals that are below in the picture are definitely longer than the 3 that are above. And you have that big hole that's opened up. And now there's basically only one axis of symmetry. You'll actually see another example of radial symmetry modified to bilateral when we talk about sea cucumbers. That's coming up in a bit. And again, the very earliest branches of the echinoderms are all extinct. They're placed in an extinct group called the Homalozoa, and we won't say much about them, except that they weren't radially symmetrical at all. There's been some controversy as to exactly how to interpret their anatomy. Some of them look almost like armored tadpoles. And there's some question as to whether those long extensions that are going off to the right on those pictures might have been tails or might have been arms. That's a can of worms or a can of echinoderms I don't particularly care to open right now. But fossils like this are part of why we still class echinoderms in the bilateria. Their ancestors and the earliest branches of the echinoderm cladogram were in fact bilaterally symmetrical. And the larvae of echinoderms are also bilaterally symmetrical. This is the larva of a sea urchin. A sea urchin egg will divide into 2, 4, 8, 16, form a hollow ball of cells, a blastula, do that gastrulation thing, start feeding once it's got a complete gut, and then start changing its shape a bit. So it goes from kind of ball-shaped into kind of prism-shaped to kind of X-wing fighter-shaped. I'm not sure how else to describe it. And ends up in this shape where you have a kind of prism-shaped body with elongated arms. And we call that stage a pluteus stage, if you're curious. And that's what a pluteus looks like. And it is, in fact, bilaterally symmetrical. The stomach, by the way, is that brownish circular thing that you see down towards the bottom of the picture. And there's tiny little spicules that are holding up those arms. That's the earliest form of the skeleton, these tiny little rods that are holding up those arm-like extensions. That's a larval starfish. It's a slightly different plan. The technical name's a Brachylaria, but you can also see that it's bilaterally symmetrical. It's not radial. Different echinoderm classes have different larval shapes and names. So, for example, sea urchins go through a pluteus stage. Brittle stars go through a slightly different type of pluteus called an ophiopluteus. Starfish go through a bipinnaria and then brachylaria stage, and sea cucumbers go through an auricularia stage, on da 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 da. They're all thought to be variations on the same basic kind of ideal type, and you see what that looks like in the middle, with a complete gut and a band of ciliated cells, which may divide into several bands or wrap around in fairly complicated ways. But yeah, all this diversity of larval types are thought to be variations on a kind of ideal that you see in the center there. But this is weird. Most of the cells of the larva don't end up as part of the adult. The adult body actually develops inside the larval body from a mass of stem cells. A mass of stem cells that's called setoside cells. So up here you've got a late sea urchin larva. It's a pluteus. You can see the little larval skeletal rods that are holding up those arms. The gut is the round thing in the middle. And then just over to the side of the gut on your right, but the larva's own left, there's a lump of cells that's beginning to organize itself into an adult body. Over there in E, you can see it developing. Those 5 little circular dots that you're seeing of the larvae are going to be the first of the adult's tube feet. So what will be the adult's body is starting to develop inside that larva. I always have a hard time kind of explaining exactly what's going on here because it can be hard to take diagrams like this and convert them into 3D. Let's just say that the left-hand divisions of the coelom enlarge, and then they wrap around the gut. And that's how you get from a bilateral larva to a radial juvenile, is the left-hand divisions of the coelom enlarge, the right ones mostly go away, and they wrap around and create instead of just being open spaces, they create these circles, which is what develops into things like the ring canal. So I'm not sure I can put it any better than that, but that's ultimately how in echinoderms you get a radial body plan from a bilateral embryo. Is the right side degenerates and the left side wraps around to form loops. Okay, I hope that was not entirely obscure. Okay, let's see. 9:45. Yeah, we'll keep on going just for a little bit. Most echinoderms are in a subphylum called the Eleutherozoa. Eleutheros happens to be ancient Greek for free, as in, not bound, not a slave, free to be you and me, free as a bird, and this bird you cannot tame. Sorry, that's the Arkansas national anthem. Eleutherozoa, free animals. These are the echinoderms that can wander around on their own. These include Asteroidea, that's the true starfish. Ophiuroidea includes brittle stars. Superficially look a little like true starfish, but very different anatomy. Echinoidea is sea urchins, sand dollars, and allies. Holothuroidea is the sea cucumbers, which is a whole nother case of radial symmetry turning into bilateral, but we'll talk about those in due time. Back around about 520 million years ago, next Thursday, there was a much greater diversity of echinoderms that had stalks and that didn't just crawl around whenever they felt like it. Many of them are kind of short-lived groups that, for reasons of time, we're just not going to be able to get into. The only group that has survived to the present day of non-free echinoderms is the Crinoidea, affectionately known as sea lilies. And even among the Crinoidea, many of them are now able to move around. There aren't very many stalked crinoids still with us. There are a few. But even most of the crinoids have evolved the ability to move around. So let's look at these crinoids before we take a break. They used to be, crinoids with stalks, used to be among the dominant marine invertebrates up until about 245 million years ago. And there's over 5,000 fossil species that are known. They used to just cover the floors of shallow oceans. In particular, the shallow ocean that used to cover, northern Arkansas, up until it finally retreated at roughly 300-odd million years ago, give or take a little bit. But if you go and poke around in the rocks of the Ozarks, more or less north of the town of Leslie, there's a lot of limestone there, and some of it contains very abundant remains of fossil crinoids, fossil sea lilies. They're very common in other parts of the U.S. that have rocks of that age. You can find a fair number of them in the limestones around places like Nashville, the limestones around places like Cincinnati. There's a lot of limestone of the right age in places like in the Midwest in general, Illinois, Ohio, Indiana's actually got some famous places to find crinoids. So they used to rule the roost. 245 million years ago, the Earth suffers the worst mass extinction of all time. May have been a combination of an enormous volcano eruption and a meteorite hitting simultaneously. There's a book on it by a guy named Peter Ward that I'll recommend if you're curious. It's called When Life Nearly Died. Something like 99% of all marine animal species were wiped out, and very few crinoids survived, and they never did quite diversify back afterwards. More on that you can learn if you get the chance in my history of life class. So there's only about 95 living stalked crinoids, mostly in deep water. About 540 species are crinoids that have lost the stalk, and we call those feather stars. So on the left, there's a fairly typical stalked crinoid. The mouth is, okay, the body is that kind of cone-shaped thing in the center. Underneath the body, you have this cup-like set of plates called the calyx, same word as a flower calyx. So the body looks kind of like a glass. And covering the glass, if you will, you've got some specialized plates held together that make a sort of roof called a tegmen. So imagine putting a napkin over a wine glass. The part that holds the wine would be the calyx, the napkin would be the tegmen, and then the stem of the wine glass would be the stalk. And the stalk is made of ossicles that stack onto each other, one on top of the other, like a stack of poker chips. Except you folks are too smart to gamble, so you don't know what those are like. How about a stack of checkers? Or a little better than that. The ossicles that make up the stalk usually have holes in the center, and there's a little thread of living tissue that holds them together. So how about a stack of lifesavers? That's the way that stalk is set up. It's a bunch of disc-shaped ossicles stacked one on top of the other, and actually nesting together because they've got grooves so that each one locks into the ones above and below it. That's the part that's like checkers, right? You know that checkers have these grooves that make it possible to stack them on each other and have the stack hold up in a rigid way. That's what stalk ossicles are like. And then the mouth is up in the middle of the tegmen. So the mouth is facing up. And surrounding the mouth, you have 5 highly branched arms. And each arm has a bunch of branches on it called pinnules. And when the crinoid extends these, they make a very efficient filter feeding basket where the pinnules trap food particles and pass them down the ambulacra, down the inside of each arm, onto the tegmen, and then into the mouth. I'll show you, I'll may be able to make that more clear in just a minute. On the right, that unstalked crinoid has all the parts that a stalked crinoid does, and unlike a true starfish, its mouth is also up on top instead of underneath in normal position. The stalk has disappeared, but there are several jointed structures called cirri. And the unstalked crinoid can actually hang on to things with those or even walk with them. I'll show you what that looks like in just a minute. There's a diagram through an arm. All of those sticky uppy things, if you'll pardon my technical jargon, are tube feet, and uniquely in crinoids, each tube feet branches into 3. They have these triple tube feet. The tube feet can snag food particles and pass them down to a ciliated groove in the upper surface of the arm called the ambulacral groove. And then food particles are just passed down that groove, ultimately over the tegmen and into the mouth. And again, stalk crinoids were quite diverse up until about 245 million years ago. That happens to be a really, really nice fossil of one. Fossils that good are rare because the thing about skeletons made of ossicles is that they tend to fall apart if you look at them funny. As soon as a crinoid died, the soft tissue would have rotted very fast, and the ossicles would have just separated. So you don't normally see fossils of crinoids that are quite this complete unless the thing managed to be buried while it was still alive or very, very soon after it was dead. So you can see the stalk over on the left, and then all of the branching arms with their pinnules on the right. The calyx is kind of in the middle and a little tricky to make out. And even though fossils this good are rare, bits and pieces of crinoid skeletons are extremely common fossils, and there are rocks that are made up of almost nothing else. That's how common they were in some places. These are all crinoid stem ossicles of different species. You can see some of them are round and some of them are, five-pointed shape, almost like little cookies or something. And in life, they would have been stacked one on top of the other, and some living tissue would have strung them all together, running through that hole. And again, you can find, if you hit the right body of rock, you can find plenty of these in many parts of the Ozarks, for example. The big gray limestones that you see if you're going up 65 towards Branson, just south of Leslie, Arkansas. If you pull over there and poke around a little bit in the limestone, you can find crinoid stem pieces the size of U.S. quarters. That's how large they sometimes get. There are still a few living stalk crinoids. Jamaica, I'm assuming that you have never seen these because they live in deep water off of the Bahamas. But you can see why they're called sea lilies. They look like flowers superficially. You can see that they've got these very long stems. The calyx is in the center and kind of buried. You can kind of see it in that middle one. But the calyx gives rise to multiple branching arms, each one with little pinnules. Incidentally, the one in the middle and the one on the right have other things tangled up in them. The one in the middle, actually, I guess that's behind it. That's a soft coral of some kind. That yellow, squiggly-looking thing is actually behind it. The one on the right, there's a couple of echinoderms called brittle stars that are kind of hanging out on the stem. So some of that stuff that looks like it's kind of tangled up in there, that's actually a separate organism. You can see how those branching arms make a filter feeding structure. You can see how the stems are long, and the stems have cirri branching off. Those little regular branches that you see are cirri. You didn't see cirri in this fossil. Not all crinoids had those, but these certainly do, those branches off the stem. And if you imagine, evolutionarily speaking, keeping the cirri but shrinking that stem shorter and shorter, you'd end up with something like this. This is an unstalked crinoid, the common name for which is a comatulid. It's got the same basic body as those stalk crinoids that you saw, but the stem has shrunk down to nothing, but the cirri remain, and comatulids can move around on their cirri. They can use them like little clumsy legs. And as here, what they're doing is just holding on to coral outcrops, just gripping onto those with those cirri and filter feeding. So they're still living in basically the same way that those deep sea crinoids are. They're just more mobile because they're not dragging around a stalk. And these are the only crinoids that you could easily see, without investing in a deep sea research submersible or something like that. Scuba divers can often spot comatulids in tropical parts of the world. You know, they're quite colorful and attractive. Worth looking for if you ever get to go scuba diving, if this damdemic ever ends. Okay, righty ho. I'm going to go ahead and stop recording and give you folks a coffee break. If anybody's got any, I don't see anything new on the chat, but if you've got some questions, I'm going to step away from the computer and refill this. So if you've got questions, you can type them or ask me when we get back. And I will now stop recording.